Hello, my name is Miwa, and today I'll be talking about cerebellum and the spinal cord. Before actually getting into the topic, let me explain how the highlights in my slides work. Red highlights are for terminologies that you must know for the exam. Orange is for terms recommended to know for ace in the exam, though not essential. And green is for terms that you do not need to memorize, but just good to keep in mind. Let's start with the layers of the cerebellar cortex. It consists of 3 layers: the outer molecular layer, the middle Purkinje layer, and the inner granular layer. Below these layers is the white matter. The molecular layer contains neuropils, which are fibrous components of neurons like axons and dendrites, along with basket cells, stellate cells, and climbing fibers. The Purkinje layer consists of Purkinje cells, which are multipolar neurons. The granular layer contains granule cells, mossy fibers, Golgi cells, type 1 and 2, and glomeruli. Granule cells have branching axons called T-axons. Glomeruli, or cerebellar islands, are masses of synapses between granule cells and mossy fibers. Here are histological slides of the cerebellum. The upper left slide is stained with impregnation, which visualizes fibers, while the right slide is stained with a regular stain. In the impregnation slide, you can observe Purkinje cells forming synapses with basket cells. Next, let's move on to the structure of the spinal cord. The dorsal horn contains the bodies of sensory neurons and protoplasmic astrocytes. The anterior horn contains the bodies of motor neurons and protoplasmic astrocytes. The central canal is filled with cerebrospinal fluid, or CSF, and is lined with cells called ependymal cells. The white matter contains axons, fibrous astrocytes, and oligodendrocytes. Now let's focus on each cell type. Motor neurons in the anterior or ventral horn tend to be large, while sensory neurons in the posterior or dorsal horn are typically smaller and round. Astrocytes are found throughout the CNS and come into 2 types: protoplasmic in gray matter and fibrous in white matter. Their roles include forming the blood-brain barrier in the brain, regulating interstitial fluid composition, providing structural support, aiding in neuronal development, replicating neurons, and forming the glial-limiting membrane. Oligodendrocytes appear pale in regular stains due to their lipid content, but they stain blue with Luxol blue, which targets phospholipids. Ependymal cells are responsible for CSF production. They have both microvilli and cilia on their apical surface and lack a basal lamina. And that wraps up the key points of the cerebellum and spinal cord. I hope you found this interesting.